Welcome, Tom. Welcome, everybody. Thank you for joining us today for our Work in Progress seminar. Today, we have with us Dr. Tom Mather from URI, who's going to talk about animal models for assessing efficacy of minimal risk natural product tick repellents. Tom Mather has very well-known reputation. I think everybody here probably knows him, but for those who don't, he's the director of Tick Encounter and a public health entomology professor at URI, which is Tick Encounter is part of the Rhode Island Cooperative Extension Program, and Tom's research and outreach activities largely focus on tick bite prevention, tick control products and strategies, and tick-borne disease education. Thanks for joining us, Tom.
Yeah, well, thanks for having me. Just start sharing my screen to make sure it still works. So hopefully we're all set there. And I want to make sure that I give much credit to Lexi White, who is a research associate here. And she's been very helpful in both putting this presentation together as well as working on the project with us. She basically shoulders all the things that I don't choose to want to do. So I'm grateful for her presence. And anyway, so I just want to make sure that she's got a fair call out. So we have had as part of our portfolio of projects when we first put in the NUVEC proposal to consider ways of assessing efficacy for minimal risk natural products, both as tick control products, and that project has completed. It was the master's student Chantal Singh Sereno that had done that, and one paper has been submitted, and just want to make sure Lars hears that word, that the paper was actually submitted. Um, and we're just waiting to hear back from the Journal of Medical Entomology now. Hopefully they like it very well. And the second one is in progress of getting submitted. So, but looking at repellents, that's another whole thing that we hadn't really thought to do initially with natural products, but then it seemed like a good extension of the whole natural product assessment work. And so we thought of trying to come up with a fairly, I'm saying this sort of tongue in cheek because it didn't end up being as simple as we thought, but a simple, sort of robust and like method for assessing repellent activity. I know that Eric here on the call today had a talk of, I don't know, probably half a year or so ago the detail that he conducted blew me away in terms of he always does with his details, but you know, of looking at repellency assays and everything. So this is a sort of a poor man's approach in a sense that we thought of doing, and it just kind of fell out from just what I've done for, I don't know, however many years I've been working on this, 40 years or so of feeding ticks on rodents. Well, I know how to do that, so why don't we just see if you treat the rodents, maybe that would be a good assay. So we didn't really know how it would work, and we decided to do it anyway. And so that's what I'm going to talk about today. So when you go in a store, you got a lot of choices. You got a lot of choices of things that say are tick repellents. And, you know, I use this, I like this slide because I'm sure that some people's motivation is looking at the price stickers underneath the products. And some people's motivation might be that they're looking for particular active ingredients in the products. And so one of the things that we've been able to think about is where are people in this whole story of tick repellents. And as sort of part of Project Scratch, that Nell and Lexi were working on for a while, and now Emily is part of that. We had a chance to talk to a lot of individuals that were already sort of thinking about preventing ticks because they were generally part of our Project Itch. So I like the fact that we can take these projects and then use elements of them to help guide and direct other things that we do. But the question is, where are you in this whole tick repellent story? And looking at what people say, I think is sometimes very interesting. Like this lady said, the bottle says it repels ticks, so I assume it does. Shouldn't I assume that? And that's a very common thing that probably all of us have heard. That people just take at face value. If it says it is, then it is, right? Or this lady, she um, said, honestly, I just feel better using something natural. I don't want to spray a bunch of chemicals where my kids and pets hang out. That's a very common thing that people say. So, let's see where, or I'm already worried about ticks. I don't need to worry about what's in the spray too. Just natural feels easier and safer. Or if I can keep ticks, oops, sorry. Uh, If I can keep ticks away without the heavy chemical stuff, why wouldn't I? Natural sprays just feel better, feel like a safer bet for me and my family. So these are very common things, and probably all of us have heard renditions of these things in terms of people going back to that shelf of products and saying, well, if if it costs money and it's there on the shelf and it says it's all natural, why wouldn't I choose that? But what happens is we don't actually know if very many of them work. Then there's, of course, the very reputable resource of trying to help people make decisions that is available online, TikTok and other recipes that people have swear by. If you know if you put this kind of information out, like in social media, it's amazing what people do. And I don't know exactly where they get their information from, but I'm sure it's a lot of these sources, probably many more of these sources than reputable sites like the Nuvec or Tick and Counter websites or other published research as well, which is somewhat limited in this whole area of minimal risk natural products in general and minimal risk natural product repellents in particular. Um, to start this, we wanted to sort of look at what other assays there were out there that people have used and how reliable are those assays in terms of providing data that would support, whether something is effective or not. And there's just a modest list of papers that have been out there looking at test systems for tick repellents. There's, you know, on host, there's on in situ papers. I can show an example of some of these. This was one from Jim Burtis and Lars at CDC, where they kind of reviewed in vitro bioassays and, sort of on skin assays. This is one that they used in some of their studies. Here's one over here that Eric described a few meetings ago. Very tech, tech, tech. He has a computer system and a camera, and he's watching the ticks and watching what they do. This one was one that I kind of made me laugh in a sense. This was a report on ABC News. Oh, sorry. You can see that I don't know how many of us apply repellents and expect it to work when it's still glistening on your skin. But this apparently in the trial that this person did on ABC News, it worked great keeping the ticks off. But I think maybe it was more that the fact that the ticks didn't like sticky, wet surfaces, perhaps. I'm not sure. Anyway, it was kind of interesting. She had gotten ticks from Tom Daniels at Fordham and then was showing this. And so she used one hand and then she used a different hand for a different product. And then she ran out of hands. And so she started using her feet. It's kind of fun to watch the report Anyway, I think as typical, we can rely on the CDC team to come up with the most authoritative information for us. And so this was a relatively recent paper in Parasites and Vectors 2024, where they looked at using one method of assessment and a whole bunch of minimal risk natural products. And you would argue that once they did that, I didn't have to do the work that we're talking about today, but we were just wondering if a different assays strategy would also be effective. And so we have a nice sort of straw man to sort of bounce some of our information off again when we get to it. But just use this finger assay, and if you're not familiar, basically they apply the treatment here, they put ticks here, and they take advantage of the fact that ticks like to walk up. Usually they do that unless someone's taking a picture or video of them, and then they don't cooperate. But um, the idea is if they get to this part, then the treatment failed, and or if they don't go across this zone at all, perhaps it worked. And then you can see data here that the deep products worked really well over an extended period of time of 5 or 6 hours. The carrier alcohol didn't repel the ticks at all. And these were sort of the data of the various minimal risk products. And you can see that generally they had some effect, at a half an hour or an hour and a half, and then the effect dropped off pretty quickly after that. So most people that, go to the trouble to apply a tick repellent are likely to be exposed for longer than an hour and a half. And yeah, and then they're probably not thinking that they need to reapply it. But whatever. So when I think about what people actually are doing, especially like with their pets, this would be a typical kind of a picture, I think. And so it was like, all right, well, we have these mice. Mice are potentially just little forms of this dog, and this is what people are doing. So why don't we just replicate that as an assay system? And so the first thing we needed to do was sort of collect some of our products. And so just thinking about it, we asked people in different areas, like, well, if you were to use a minimal risk natural product, what's your favorite repellent? And so we got a lot of people that were fans of Nantucket Spider here, or Yaya. Yaya is big on Long Island, Lexi says. A lot of people swear by Wonderside, and then Cedarside. People seem to like cedar products. I think it works well. It probably makes their dog smell nice. And then our friends at Evolva, now no longer Evolva, but had their Nuctitone related products as well that were being developed as repellents. And then comparing them with, two fairly standard products, picaridin and DEET. So that was going to be our plan for this study. And just moving forward from that, we decided that we would look at sort of a simple toxicity assay on our animal model and then an efficacy assessment as well. And you don't have to read all of these words, but basically, we just wanted to make sure that treating the mice didn't cause an adverse event or outcome. And so we sort of went through the procedure of what we were going to do and just watched them over the course of a week or 2 weeks. And some of this is still ongoing. And we have some of our undergraduate student interns helping with making these observations, just checking in on the mice and making sure that they don't turn red or their eyes don't pop out or anything like that. And then hopefully applying it with a study with ticks as well, and just looking at what that looks like. So this mouse has been sprayed. We shaved some skin some hair off so that we could sort of watch whether there's any redness that develops in the skin as well. And so that basically is where we started. So this is a work in progress. And so this is about the progress that we've made so far. We've been working through and are just finishing up sort of the toxicity assessments for the products that we had. We had charged ahead, several months ago, but I'll get to that, what the outcome of some of that was in a few minutes. So we've gone back now and sort of have assessed our 7 or so products from a toxicity standpoint. And then moving forward, we will take this and apply it with 2 time points, sort of apply it and let the mice sort of dry off for 30 minutes and also for 4 hours. So we'll have two of the time points. The CDC's work had several time points that range between a half an hour and 6.5 hours, I think. So hitting sort of a happy medium with 4 hours that allows us to get the work done in one day, I guess, without spending overtime. I don't know how the CDC team figured that all out, but this is just what we're planning to do going forward. So once the mice have reached sort of either the 30-minute or 4 hour time point, then we can challenge them with 30 nymphal stage black-legged ticks that we can grow in the laboratory. And once we have these little cages that contain the mice, we can put them in the cages and expose them to the ticks. And then what we would do typically would wrap them up in paper towels and let them sort of have an hour or so of sort of opportunity for the ticks to either get on the mice or not. If the mice aren't treated, generally speaking, we have a very efficient, if we put 30 ticks in, we'd expect somewhere around 28 ticks to be attached, and maybe a couple would, not attach after an hour or so, but it's a pretty efficient way of exposing rodents, ticks, ticks on rodents. Um, so that's just our plan, how we're going to proceed. There's been a few modifications that have come up. And I'll talk about that in a second. But one of the things when we put animals in, we have a new campus vet. She wasn't very keen on exactly what we've done for 40 years. And so she, having never actually worked with ticks at all, she decided that we should try and think about modifications of our protocol. So, of course, that's fine. First, you have to think of the modification and implement it. And then you have to, of course, before you can implement it, you have to change all of your IACUC protocols, and that takes time. So, anyway, I'm going to sound like a sour grapes person here for a minute, but um, it has made something that seemingly was a no-brainer kind of a process into a far more complicated process than I feel it needs to be. But now we have, wired these cages so we can monitor the temperature to make sure that everybody's happy. The idea of using stocking net, you can see there's more pores in it than paper towels, so perhaps that will allow um, a better degassing of the repellent defect, maybe. And this just shows sort of how we would lay out our data, give you a sense. You can see when we did just some initial trials with just DI water, we had, I'd say, a relatively poor compared with our normal success when we're using Peromyscus mice and ticks that our recovery wasn't as high as I expected it to be, but it's reasonable, and we should be able to see differences when we start doing this with products. So we did start doing it with some products. And one thing that happened was we ended up fairly quickly with some adverse reactions that, of course, never makes anybody happy. Clearly didn't make Lexi or I happy, nor the new campus vet. And some of the things that have sort of caused this project to run off of its track a little bit were these adverse reactions. And they're trying to figure out why that was because the toxicity part of the study seemed like it was just fine. And so what was going on with this? So one of the things that happened for us, and it happens in animal work all the time, that sometimes the methods that you used, in this case, the drugs that we had always used to anesthetize the animals, were not available. And so we had to make slightly informed choices of alternative drugs. But of course, we had used those drugs. We've used xylazine um, as a drug to knock the mice out for years, and then we had to switch to a couple of other options. And we didn't really know all of the parameters that generally worked without adverse reactions. But in retrospect, we had more adverse reactions than I ever remember having when we had used xylazine and ketamine. So happily now we are able to get xylazine again. I'm not exactly sure what happened. I guess xylazine became kind of a street drug for a while, and it made it a little bit hard to get from a regulatory thing. And we thought we wouldn't get it back, but right now at least we have a supply of it back. So hopefully that's going to be one thing that helps us moving forward, that we don't have to deal with drugs that we're not exactly sure what might be happening. Now, of course, the other thing that we were doing differently, we were applying something to these mice, these repellents. When we did it in a toxicity test, it didn't seem like it had any problem, but we had a complication. The way the protocol was going to work, we were going to knock them out with isoflurane and then treat them with the repellent and then let them wake up. So that way we didn't have to inject xylazine and ketamine into them more than one time. And we like to do that before we put the ticks on. So that could have been the isoflurane system that we were using was causing a complication. We hadn't really sort of, there might have been an interaction between that. We've now decided after some discussions with the campus vet that we can knock the animals out with the xylazine and they stay out for about an hour. So that covers the 30-minute wait. We would have to give them a second dose if we're doing the 4 hour treatment. And so that's an issue that we've just had to go back and forth with. And anytime you go back and forth with the campus vet, it always takes a little longer than you'd like it to. But that's, I think, where we are right now with this. But we've also sort of done what she wanted us to do. We changed the fabric container, the mesh, maybe allow a little bit more airflow, and we are monitoring the temperature of the mice while we have them caged. And we also have reduced the number of sprays. So when we were spraying them, the first protocol was spray them till they were wetted. That seemed to make sense. That's what was being happening on like that dog in the picture earlier. Um, but 10 maybe is too much, and so we've cut it back now to 5. So as part of our little detours thing here, we've had to make these changes. So of course that's caused us to have to make protocol modifications and get them reapproved by the IACUC and everything. And so while we were busy doing that, why say we, but it was really Lexi that was charged with all of this. She's probably sick of changing and modifying protocols. We added a few additional repellents as well. So instead of just the Nantucket spider and ya, we added these Wondercide and Cedarside products as well to the mix. And we'd be happy in the discussion to hear if there are other products that we might want to consider as a second round once we get this going and get some initial data if people have other favored products out there. I know I've spoken with Eric about some other opportunities. He's had some interesting work that he's been doing, and maybe we could try that on this system as well as the systems that he used to assess their efficacy. So with that, I'm happy to answer any questions, or we could have a more of a discussion about this whole idea of, you know, what people, what should people do when it comes to thinking about preventing tick bites using repellents, both on their pets and on themselves. I'd be happy to have a discussion about that. So, Matthew.
Hi, Tom. I want to ask a question about the presentation. I saw that you're looking at both toxicology and effectiveness or efficacy, and it seemed like you had a table that was about to show more efficacy. Are we not quite at that level yet? We're still at the tox level, or do you have information you can share?
Yeah, we don't. It's actually one of the reasons I pushed this. I switched with Steve because we have no data yet. It's a work in
Okay.
progress. So we had some trials, not counting them because more than half of the mice died in those trials.
Mm-hmm.
So we're not really trusting of that. So I kind of threw those out. Hopefully, when we get going again with this new protocol and the new drug scheme, that we won't have that problem. Um, yeah. So
Okay. Well, let me do a follow-up question, seeing we don't have that. Um, let's talk about kind of what drives the desire for natural products among people. I know you've done a lot of work with this, and you've done some testing, I believe, in the past at University of Rhode Island. What's going on with the public and the idea of natural products? You did some nice quotes at the beginning, trying to reach out and connect with people as you do well. Um, how much is this the basic thing we've understand for years of kind of chemophobia, that most people don't want to take what they appears to be a synthetic chemical if they have an alternative of a natural. Clearly, that's you know subjective, people's perception of that. But when you talk to people, what do you hear from them about more about why they, accept one over the other? Is it go any deeper? Or is it just, currently the kind of the general feedback you get that you showed in the earlier slides?
yeah, that's a great point, Matthew. It's something that we think and talk about it with our tick encounter program a lot. I think that those messages that people said, those are people's actual words that Lexi recorded when she was talking with them, generally reflect sort of the thought, but maybe others on the call have some
Mm-hmm.
thoughts too. I think, you know, why wouldn't you use natural? And it's the same with Project Itch. People, we often hear, when they are making selections for what they're treating their yards with, well,
Yeah.
I'd rather have a natural product for the same basic reasons of not having something that their perception is that the synthetic products are toxic, and they're toxic to more than just the
Mm-hmm.
ticks, right? And
Yeah.
so what I think is missed in all of that myself is that if a natural product is going to kill a tick or repel a tick, it, there's an active going on there, and I think it's maybe short-sighted in a way to think that it's not having some effect. I mean, clearly our adverse effects with our mice, there was something, maybe it was the drugs that they didn't like rather than the repellents, but or the interaction of them. But um, yeah, I think the natural products, just because they have the word natural, doesn't mean that they don't have some kind of effect, right?
Sure.
I
There's
mean,
there's
if
always an effect or side effect.
my just crudely thinking, I often think to myself, well, you get this natural product and it says it's a natural tick killer. Well, it's not selective. If it's going to kill a tick, it's probably going to kill anything that it hits when you're
Mm-hmm.
spraying it in someone's yard. But the way it's marketed is, people read those words and that's what they believe, right? And so
Yeah.
I don't know. Other people may want to weigh in on their thoughts about this. I'd like to hear.
Steve, you have your hand raised.
Oh, I don't want to take away from that stream because I have a different question. So,
Yeah,
In
does
that
any?
same thread, I'll interject really quickly. I just wanted to emphasize like the things that we talked about throughout Steve's talk or Johanna's talk, right? Leaning into this idea of literacy around these things. Folks hear natural, and that's a word we understand. And of course, we want natural, right? Especially if you're an environmentally driven human. But it's always interesting to me that synthetic is a word where we're like, that's not natural. And that's most of the literacy that I hear. But if I break down even permethrin down to chrysanthemums and like it's highly specialized, right? I get a greater acceptance from homeowners than here's this synthetic chemical that works really well. They just hear synthetic. But I think that's a big field in terms of literacy of navigating. Yes, it's a chemical, but everything is. Um, and getting rid of the fear around that and more of like an awareness or empowerment, I think is a huge, huge piece of this. And all of the aspects, I think, in NUVEC especially, are really getting at targeting this in different angles, including Project Scratch. So,
Just in follow up, I'm trained in behavioral sciences and public health, and I'm always trained to respect the fact that people are good consumers and they're good at doing risk assessment for themselves. And I'm kind of like, I want to use that information and try and understand better why, when someone selects a product, they look at the advertising, natural versus synthetic, but I would think they'd also really want to know about effectiveness because they're just good at, they want to choose the thing that works best for them for the money. So that'll be a longer conversation, and I'll try to follow up with Tom and maybe Alexis to talk about that just to wrap up this phase and move on. But I think we have to consider more about how we talk to people as consumers rather than just as scientists, as logical people. So
Yeah,
thank you, Tom
I
and Alexis.
appreciate that thought, Matthew, and yeah, I'd love to continue talking about it. What we have found with Project Ditch, at least on our side,
Hmm.
is that we often will hear, well, I use, and they say a natural product. I don't know if it works. Maybe it doesn't. But they're still using it. And
Yeah.
it's like at some point there in that whole mix, people sort of, I don't know if it works. Well, we're telling you it didn't work in Project Itch.
Yes.
And we have seen, and it would be interesting for us to do that, Lexi, would be to say how many of the people that we gave results to and then
Hmm.
come back the next year and we've included them again because they've now switched they've either gone from a natural product to a treated product. I just there's a couple anecdotes in that, and one was there was this one family the first year they they were kind of on the fence whether they did treatment or not, but they said they did, so we included them as a treatment. But they apparently used sort of a natural product, but their yard happened to have 6 or 7 Powassan infected ticks. And the next
Hmm.
year, they were strident about the fact they were going to switch to something that maybe worked a little bit more. Um, they still only made a half-hearted effort in that because so they asked me who would I use, and I suggested somebody that had a truck-mounted sprayer that sprayed bifenthrin, and they ended up with Mosquito Joe or whatever that had a backpack sprayer and permethrin rather than bifenthrin. So they didn't quite follow. At first they were going to do exactly what I said, and then they ended up doing something slightly different. And actually, we were there once, and I was watching Mosquito Joe spray, and what we found in their yard was that we have a zone A and a zone B, and zone A is right at the perimeter of the lawn. That they did have fewer ticks in zone A, but they had not so many fewer ticks in zone B. And the guy was spraying because he's Mosquito Joe. He's spraying for ticks and mosquitoes, and he spent more of his time spraying bushes, 5 feet and higher rather than the ground. So, it comes down to the fact that the application of any of these products also probably has an impact as to in terms of the efficacy of them, at least from a yard spray. And it may be the same case with the repellents. I think, most people, when I watch them, I give them a can of spray and I watch them and they do a little sit, sit, sit spray, a little bit. And that might
Hmm.
not be really enough if talk to the people that make permethrin treatment for clothing, they tell you that it needs to be wetted. Well, most people never spray that much on their clothes. So that's going to impact efficacy probably.
Thank you. Very helpful examples. I'll follow up with you.
Yeah, great.
I wonder, Tom, if you think about the endpoints of the repellency, like what's going to measure the efficacy? Like, is it attachment? Is it whether the tick gets on the animals,
Yeah,
or
so the
whether
way
they
we've
feed
set it,
fully to completion? Or
yeah, the way we've set it up was that we monitor the end point for us is how many of those ticks actually went ahead and bit the animal and take a blood meal. We assume that if they bite it, it's likely to eventually turn into a blood meal. And so we actually put them in cages over pans of water and harvest the ticks out. But we monitor that on a daily basis. And we do get, after the first day, we get some that just weren't really attached and they end up in the water unfed. But mostly I think the data that we're likely to be reporting is how many fed ticks do we recover from those animals?
And as a follow-up question, can you imagine where that repellents could be used in not necessarily as a personal protection for humans, but as a measure? Because then they could be seen as specific. You're preventing ticks from biting hosts in the wild, as opposed to killing ticks in the wild. That
Yeah, why not? That seems reasonable. Some of the products, though, are that we're testing are designed as repellents, and so people are expecting it to be you know, I mean, you're talking about sort of an endpoint. I have to give that a little bit more thought, I guess, but
I would be
I always,
interested in following up on that too, because we have a lot of folks here who use repellents for animals as tick control. So like keeping deer and rabbits out of their yard. I found was a common theme here that I hadn't seen in other places that I had worked. Um, but it's just interesting, again, another, not particularly a tick repellent, but maybe if it repels hosts.
And just one thing about the chemical stuff, I think it's a lot of that is the ghosts of Rachel Carson, right? So, and one way that I counter people when they say they want something natural, they say, well, ticks are natural. Do you want ticks?
yeah. So one of the things I always say is people are, I want to use a natural product. And, I'm thinking to myself, yeah, but some of these things may or may not be as innocuous as you think they are. I think Matthew's got, a good point to follow up on that, just how it's framed and how people, what people's reception of it is. I don't think I've hit on the right answer yet in terms of the communication about that. It's probably an age-old problem. Definitely something that I believe, you know, we should spend a lot more time thinking about those. So maybe that's something that, we can develop like a little group within NUVEC that wants to talk about some of these things. So Lars, you wanted to say something?
I just wanted to spin back to the stuff that James Curtis was doing, leading a little bit in terms of, oh, to me, that there are two kinds of repellent products for ticks. There are EPA registered repellents and non-EPA registered repellents. And to me, the very clear benefit of the EPA registered repellents that you can buy off the shelf is that in addition to being considered safe for use by EPA, they also have a requirement for efficacy. And EPA has a standardized human skin bioassay. That's the one that Tom showed a picture of, where basically ticks get put on the forearm. They call it the forearm assay. And that is the standard method that they use to determine that the repellent is efficacious enough to be approved. And you can see James's slightly hairy arm there. So then you basically have the actual product applied to a human skin surface, and you introduce the ticks and determine to what extent they're repelled. And the products that are on the market are usually tested against an Ixodes tick, an amblyoma tick, and a dermacenter tick. So those products, you pick them off, you can be pretty sure that they work, and they work for as long as the label says. When we get to the 25B minimal risk exempt products, they're all safe, we assume. And I think that's a fair assumption that they are safe, but efficacy is wildly variable, as James showed in the paper where he used the simplified finger assays. You can see they all worked to some extent for the first 30-minute time point, and then they absolutely crashed. So I think one thing here is you could get pretty good protection with some of these products, but you have to keep reapplying them probably every 30 minutes. And that may not be clear to people. That's not something you would find on the label of a bottle you pick up in the store. So there's a problem there with the 25B products that are not registered by EPA, which also means that they're easier to produce and market because you don't have to go through a regulatory process. But it's the buyer beware thing. You don't really know if you're protected or not. And it's not clear from the products how long you're protected, how often you need to reapply. So the other thing here is that there is a wild proliferation of these 25B repellent products. And there is no entity that is responsible for testing them to see if they are effective or not. There's no regulation. And we tried a few, but to try to keep up with doing that in a way that you test everything that's on the market, you have to keep testing what's on the market, is a monstrous task if you want to keep consumers up to date on what's on the market, what's effective. And yeah, there's no one doing that. And I can't see that anyone will be taking up that torch either. So makes it pretty tricky from a communication standpoint to put those messages through in a way that people understand and would be accepting of. I mean, to me, if you had a natural product that was actually registered by EPA, you'd have it both ways, but not many companies is going to go through that regulatory process unless they have to. Because that's a lot of cost, it's a lot of time, it's a lot of effort. So this is similar to the acaricides that go on the ground to kill ticks. This is it's a big thorny problem. And just Tom, in terms of James used the treated skin, the finger assay, because it's easier to use, it's quicker, it's easier to keep track of the tick. So it does basically the same thing as the EPA registered, the EPA process for that they use for that registration, but it's much quicker, it's easier, and you don't have all the hair on your arms to deal with. So and you do have the human skin surface, and it works pretty well. And as a benefit too, we did not have to anesthetize James when we did those assays. So
Yeah.
he was fully awake the whole time. And I think it's pretty quick to do. And even in the first paper you showed, James tried to look at how that assay related back to a Petri dish assay to see if the Petri dish assay gave basically the same results. That would be an option for quick screening. So I think that your mouse model is somewhere in between of those two. If you don't want to use people, but you don't really trust the Petri dish assay. So an animal model is kind of a middle of the road thing, and then it just becomes a matter of how hard and complicated it becomes to run that assay with the mice too.
Yeah, so I didn't, when I first came up with the idea, I didn't think it would be hard because it was the thing that I've done for 40 years. And then all of a sudden it was hard. And we didn't exactly know why it was hard, but yeah, it's been a little bit frustrating for both Lexi and I.
So did you guys? I don't know, maybe I missed it, but was there is not really a time point aspect to this because the repellent goes on and then the ticks go on, and they don't necessarily go on at different time points after application.
Yes, they do.
So
30 minutes
30 minutes
or 4 hours.
or 4. Okay, so that's 2 time points. So to
Yeah.
see if it's still active.
We
Okay.
haven't done that yet, but once we get our whole system so that it doesn't have adverse events, then our plan is that we would move it along that way.
Yeah, I mean that's another thing too. I mean, I think James did 30 minutes, an hour and a half, 2.5 hours, 3.5 hours, 4, 5.5, 6.5. So you kind of lose the information there between 30 minutes and 4 hours too. If I mean
Yeah.
you can't really it'd be crazy to the amount of animals needed if you wanted to do more time points than half an hour and 4 hours. So that's
Right.
yeah.
We thought that that was a reasonable thing. I knew that you all had done, those hourly time points out,
Yeah.
but
So I think
that's
I
way.
think if you look at those curves, the first time point's 30 minutes and it looks pretty decent for most everything, but by an hour and a half, it is crashing down to basement level for a bunch of them already. So
Yeah. Right. So that's why 4 hours seemed like it would, you know, everything might work when it's still wet on the skin, like
yeah.
the ABC study did. But once it's dried and volatilized off, maybe it wouldn't work as well.
It's been
Anyway,
interesting too to see how many we get that go like and feed to repletion on the animals as well, right? So like we get the initial repellency at 3 and 4 hours, but then there's also some level of like, I don't know how we'll extrapolate it to be like disease risk, right? And that factor, but just seeing how many of these ticks we're actually getting back that are able to move on to the next life stage, despite the repellent, I think is an added piece of the animal model at least. But yes, lots of detours.
so I think a good outcome here is that we have a group that wants to think about sort of how to communicate all of this a little bit more effectively and efficiently. So I look forward to participating in that. It'll be interesting to see how many of James's products are. I don't know if you did the same thing we did and just looked at what is out on the shelf, but I thought about asking all of the co-Is what their favorite natural repellent was or in their area, like we did with like the Yaya. But it was interesting between, I think Nita loved Nantucket Spider and Lexi liked Yaya, and we had both of those, and even though they're supposedly the same products, they definitely, when you apply them to mice, apparently it's quite a difference in terms of potency, smelling, and I don't
Yeah, I
know.
think James went on Amazon and Target and Walmart websites and tried to pick out the ones that were being used most frequently based on information from those types of websites. So it's and
Yeah.
it's a little bit tricky and sensitive this too, because you test actual products, you present results for actual products on the shelf, and you're not testing all products on the market. So I know I know that Kirby had some issues with that in terms of acaricides to kill ticks, in terms of companies not being happy with the results that
Oh
were
yeah.
published. And it can be construed as being unfair if you test a small fraction of what's on the market and you find something that's not working and that's published, but you haven't tested everything. So there's a little bit of an aspect of that in this too when you present these kinds of data.
Yeah. Something that researchers don't like to have to think about getting, cease and desist orders of things, with the results of their studies. Anyway, well, thanks for the opportunity to share and for all of your comments.
Thank you, Tom and Lexi, for your work on this. Yeah, in 2 weeks, we will have the next work in progress, and that will be with Emily Burton from URI on December 3rd. So thank you, everyone, for joining us.